Hi everyone, I'm Goldy Singh from Amrita School of Biotechnology, currently doing my MC Biotechnology. So I'm going to talk about metagenomics and neurodegenerative diseases. So this paper discusses the use of metagenomics, next generation sequencing as a new approach to study neurodegenerative diseases such as epilepsy, AD, PD, sequencing the genomes of pathogens. We also find the association between the gut microbiota and the environmental factors like diet, antibiotics, toxins, and how to understand the impact on host health and that can identify risk factors for diseases. Our objective was to how to use the metatranscriptomics that can provide a plethora of information of gene expression of microbial population to identify their genetic functional material in the gut microbiome. We also provide an overview of the tools and methods for studying the gut microbiota in neurodegenerative diseases and presents a case study in this emerging field. So, what are the roles of gut microbiota in neurodegenerative diseases? Whenever there is a disbalance in our gut microbiota, which refers to the term of dysbiosis, there are a lot of modulations in immune system that can lead to the production of neurotransmitters and regulation of the blood-brain barrier. The gut microbiome of individual with like Parkinson's disease was compared like whenever there is a dysbiosis, it can lead to Alzheimer's disease, Parkinson's disease, and depression, stress, which can cause to a lot of different neurodevelopmental disorders. So our pilot case study was on Parkinson's disease. The metagenomic data sets of patients of Parkinson's disease, two samples were taken from various parts of the United States and sequenced to understand the role of gut microbiota in neurological implications. So total 524 PD patients and 316 controls were taken. Stool samples were taken. After that, we did 16 sRNA amplicons. It was seen that people with the Parkinson's disease had overgrowth of opportunistic pathogens, their gut microbiome, which could contribute to the disease genesis and progression. The healthy individuals were compared with the Parkinson's disease. Another study is that a total 500 samples of PD were taken. For this, we used Velvet and MetaBait2 were employed for the data analysis, and we investigated the association between the gut microbiota and the Parkinson's disease to examine the correlation between PD, epilepsy on the gut microbiota from 500 samples. Our results was that we found that an excess of opportunities infections were discovered in PD in our case study, but we did not find any strong association between the epilepsy and the gut microbiota based on our analysis. Further, we can conclude that if we can explore the association between gut dysbiosis and neurodegenerative diseases such as epilepsy and all other, we can investigate different biomarkers, therapeutic interventions that can target the gut microbiome. And the integration between the metatranscriptomic and metagenomics can enhancedly provide the understanding between functional genetic material for the further studies. Thank you.